So, what is embryology? Embryology is the study of the origin and development of a single individual, okay? In humans, we call this pregnancy. In all things, we call this pregnancy, but okay? We split up prenatal development into two phases. The first is the embryonic period, which is the first 8 weeks of development. So much stuff happens in those first 8 weeks, you would be shocked. And then fetal development is the fetal period is the remaining 30 to 32 weeks. Okay? And we're going to go through most of the things that happen in humans, 40 weeks of pregnancy, approximately. Some mammals have a longer gestation time. For example, elephants are pregnant for about 11 months as opposed to our 8 to 9. So if you see a baby elephant out in the world, be very grateful. Those are not super common. Okay, cool. Let's do it. So the prenatal period, the first 8 weeks of the embryonic period, starts with fertilization. And about 3 weeks in, we will have developed what we call the embryo. Before this point, there's a bunch of other names that the embryo, the zygote, gets, and we'll talk about all of those as well. All that stuff, okay? Eventually, you'll hit that 5-week mark. In that 5-week mark, you'll have some other things start to develop. Your spinal cord, your brain starts to kind of form up, your heart, all of your other internal organs. And then around 8 weeks, you lose your tail and you continue forming and developing all of your organs. And we'll get into the details of all of that in a minute. And then after that embryonic period, we move into the fetal period. At this point in time, 12 weeks and up, we call the thing a fetus. I will refer to it as several different names. You'll probably hear me call it a naked alien baby or a raw chicken because that's what they look like. But again, we'll get into that. Okay, basic body plan. As we are developing in utero, your skin, which is the dermis and epidermis, that forms the outer body wall, the inner body digestive system, the kidneys, the gonads, the limbs, they all start to form kind of in utero from these different cell types that we call germ layers. And we'll get into the formation of those germ layers in a minute. Okay, remember something that I said at the very beginning of the class? Humans have a tube in a tube kind of system. This is where that comes from. Okay, this is a highly simplified version of this tube within a tube system. Okay, it won't let me zoom in. Okay, but so the blue is the outer tube. All of the red ends up being like muscles and organs and all the in-between stuff. And then the yellow is another tube, and that one tends to be your digestive tube. So the outer tube is your skin. The inner tube is your digestive system. Simplified. Do y'all remember my little thing about the asterisks with biology? This is kind of one of those things. What I have just said is the very simplified version. The inner tube is your digestive, the outer tube is your skin. That is a highly simplified verbiage of what's actually going on here. Okay? Cool? You with me so far? Great. So, week 1, going from being a zygote to a blastocyst. Let's first define a zygote. A zygote is one sex cell from one parent plus another sex cell from the other parent that makes this zygote. Okay? Each of these sex cells has n number of chromosomes. It is haploid. N equals haploid. 2n equals diploid. So when these two haploid sex cells come together and fertilize, the sperm will fertilize the egg, that is them coming together, and form a zygote. That zygote will have a diploid. It will have 2n amount of chromosomes. Okay, does that make sense? So you get half of your genetic information from your mom, you get half of your genetic information from your dad, from your male biological parent and your female biological parent. Okay, you with me? Cool. So after the zygote, that's what's supposed to go here, is fertilized, it starts to move towards the uterus. Okay? And then it also starts to cleave. That cleavage is just those cells starting to do mitosis, chapter 3. Mitosis, we know what mitosis is, right? We talked about that. It was over there and over there. It's the process of making new cells, making more of the same cells. Okay? So they're going through mitosis. They're dividing. And at around day 3, we form a solid cluster of 12 to 16 cells that we call a morula. Okay? A morula. That morula will continue dividing. And that at around maybe like the end-ish of day 4, we start to form a blastocyst. A blastocyst is the same morula, but it's just a lot more cells, so like around 60 or so, and it's full of fluid. So we started with our one cell. That guy divided into a bunch of cells. Let's go with 4-ish. And then eventually we form that blastocyst, which is a bunch of cells, and it's filled with liquid. Can you see that yellow? That is the liquid. Okay? From that blastocyst forms the inner cell mass. That's supposed to be an E. I'm sorry. Bro. I-N-N-E-R. Inner cell mass. Can't fit. Yeah, inner cell mass. And the trophoblast. The inner cell mass is eventually what will become the embryo, will become the fetus, will become the living, breathing person eventually. The trophoblast is the placenta. That is just the nutrient tube that gives nutrients to the fetus from the mother. Okay? Cool. And so it kind of looks something like this. So this outer layer of cells, this will become the trophoblast. And then all of these cells on the inner side are the inner cell mass that will become the fetus. I'm going to start abbreviating inner cell mass as ICM. Takes too long to write that whole set of words. Okay? Cool. So, like I said, in that first week, we have the zygote. That's the egg plus sperm makes zygote. Eventually you'll get to the 4-cell stage. That happens. Once you get to the 4-cell stage, you continue dividing, and you have a morula. That's about 12 to 16 cells. The early blastocyst has some fluid and separates into ICM and trophoblast. And then the late blastocyst will start implanting into the uterine wall at this stage. Bro. Okay? So that's week 1. That's week 1. This is a uterus. Okay. Okay. Cool. Cool. Visualization of the words that I just said. Egg starts in the ovaries, gets pushed out. It's actually really, if you ever get the chance to internally watch an egg be released from an ovary, it's crazy. It looks like a rocket just shooting out of that thing. It's yucky. It's not yucky, it's cool. The human body's awesome. Okay, anyways. So we move on, we go through the stages, blah, 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 blah. We'll talk about twins later. Okay, week 2. Yay, we finally got into week 2. We're a bunch of cells just living our best lives. Great. At the beginning of week 2, we form what we call a bilaminar embryonic disc. Okay. That bilaminar embryonic disc is just the inner cell mass, bless you, that separates into kind of 2 layers of cells. I don't like that color. Let's do 1 layer be red. That first layer is called the epiblast, and the second layer is called the hypoblast. Okay? And those 2 layers together make up that bilaminar disc. It's this specific little part in between where the 2 kind of join together. Okay? The amniotic sac is formed from an extension of that epiblast. So the epiblast kind of splits itself even further. Okay. And then the outer cells will become the amnion. The amnion is just the fluid that surrounds the fetus so that it is protected. When you hear, oh, my water broke, it's that amniotic fluid that is being released, that is popped or broken. And then the inner membrane forms the amniotic sac cavity. And that's where the actual fluid gets filled in. Okay? The hypoblast also forms stuff It forms the yolk sac. The yolk sac will eventually turn into the digestive tubing, right? It will eventually become the inner tube that is your entire digestive system, but it is not a source of nutrients for the embryo itself. Like in a chicken egg, for example, the egg yolk ends up being like a significant source of energy for the growing chicken fetus, but that's not the case in humans. Humans get all their nutrients from their mother. Okay? The tissues around that yolk sac do give rise to some of your early blood vessels and blood cells. Okay? And that yolk sac is formed by the hypoblast. Yoey. Epiblast. I'm trying to keep my colors consistent for you guys. Epiblast, hypoblast. All of these loops that I'm making, these are cells. These are cells that are, because in all of the time that this is happening, these cells have not stopped dividing. They're still continually dividing, making more of themselves, blah, 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 blah. All of that jazz. Okay? Cool. Here's a little visual. Around day 4, day 5, blastocyst is kind of still floating. Day 6, it starts to really adhere to that cell wall, or the blastocyst will really start adhering to the cell wall. At around day 9 is when you get that split of the inner cell mass. Okay, so day 7, we have the trophoblast doing its thing, starting to form, become that placenta, and then the inner cell mass starts to form, starts to split into the epiblast and the hypoblast. So that the blue here is the epiblast, the yellow is the hypoblast. Okay? As we continue going on through that development in that first couple of weeks, we see the generation of that bilaminar embryonic disc. That's the mix of the epiblast layer and the hypoblast layer. Hypo. Okay. And the trophoblast continues to grow into the placenta so that that fetus can get nutrients. Right now, it's taking all of its nutrients, because it has implanted itself in the uterine wall, it's taking all of its nutrients from the uterine wall. But that's not sustainable. So it needs to build the placenta in order to get nutrients from the mother's blood system. And that's why you'll see people like take their placenta home and like cut it up and make it into pills and all kinds of stuff, because it is super rich in blood vessels, in nutrients, in all kinds of stuff. It's actually pretty good for you. It's just a little tricky to think about. Okay. Week 3. Woohoo! So at the beginning of week 3, that's when we start to transition from being bilaminar to being trilaminar. You saw kind of at the very beginning with the body plan, there were 3 layers. So we've dealt with the first inner tube, that's the endoderm, and the outer tube, that's the epiderm. Now we need to talk about how we develop the mesoderm. Okay? But before we get there, the primitive streak is a raised groove on the dorsal surface of the epiblast. What side is dorsal, the front or the back? The back, yeah. So that primitive streak is going to show up on the back side of that embryo, okay? And then in a process that we call gastrulation, the epiblast cells kind of bend in on themselves. That's what that invagination means, okay? So those cells kind of bend in on each other and start to fill in kind of the space in the middle. Let's talking about. Those 3 germ layers are the endoderm, Endoderm will eventually replace that hypoblast. The mesoderm, bro. There's an R and an M there. You see that I'm trying to write it. It keeps saying it doesn't want it there. That's not my fault. I'm doing what I can. That's supposed to say mesoderm. Okay? Yeah, hell yeah. And then the last one is the ectoderm. And those are formed from the epiblast cells. Bless you. So when all those epiblast cells are doing that invagination, they have to push some of those hypoblast cells out of the way. And that is part of the process. All layers, endoderm, mesoderm, ectoderm, they're all derived from the epiblast. The hypoblast was at one point the epiblast, remember? It was all one thing and then it split. So all of your germ layers are formed from the epiblast. Caller. I don't know who you are. I'm in class. Okay? And that's kind of what we're running with here. Again, this is that kind of 3D-ish image. I can't really zoom in on here, but remember the kind of thing that I showed you in my notes. You have the primitive streak, the cells that are invaginating, and then you form that mesodermal layer. The endoderm gets pushed down a little bit, and the ectoderm kind of stays on top because it's the epiblast. Okay? The notochord is incredibly useful for defining the body's axis. It tells you what is on the left and what is on the right. It splits the body into that bilateralness that makes us chordates. Okay? At one end of the primitive streak is the primitive node, and the primitive node is what will eventually start to form that notochord. Okay? It is eventually where your vertebral column will be, and it shows up around day 16-ish. Okay? Kind of goes up. It's here. Notochord goes up and it goes down. Okay, don't worry about that too much. Just know that the notochord will eventually be where your vertebral column forms from. Okay? Cool. Visual to the words that I was just saying. The split guy does its thing at the primitive node. Remember, that is just the swelling at the end of the primitive streak. The notochord starts to form, and it'll form going up and going down towards the head and to the tail. Notochord spans the entire length of the embryo at some point, right? Okay? You with me? This is where it starts to get a little hairy, okay? Neurulation. Neurulation is where the ectoderm, so the outermost layer, starts to form your brain and spinal cord. Okay. The ectoderm will also form your skin. Okay. So brain, spinal cord, skin is going to be formed by your ectoderm. How does your brain and spinal cord start to form? That primitive streak the primitive streak that became that notochord will eventually, the notochord, notochord sends signals to the ectoderm to start forming that brain and spinal cord. It will form a neural plate in the dorsal midline. So in the middle of the back, it will start to form like a little bump, a little plate. Okay? That neural plate will become a neural groove. Plate, groove will become a groove, and then that groove will become a tube. Tube. Plate, groove, tube. Plate, neural groove. It actually grooves this way, but neural groove, and then neural tube. Okay? Once you have your plate, groove, and tube formed out, the cranial part will eventually become the brain, and the rest of it will become the spinal cord. All made of ectoderm, which is made from what cell? The neural crest forms your sensory nerve cells. Okay, so all of the nerves that deal with like smelling and taste and touch reception and any kind of sensory information, those are formed by the neural crest, specifically the neural crest cells. Neural crest cells are great. They do a whole bunch of stuff. They'll also become ganglia and melanocytes. Okay. How do these cells know what to become, what to do, where to go, how to be? Through induction. Induction is the ability for a group of cells to influence another group of cells. So when our epiblast cells were invaginating, right, and becoming those kind of mesodermal cells, the first set of epiblast cells said, hey, I need you, group of cells over here, I need you to start moving so that you can become the mesoderm. I need you to start moving so that you can become the endoderm. I need you to start moving so that you can become a heart and that you can become whatever. So the cells that are nearby another cell can have an effect on that cell. Because all of these cells started as stem cells. And stem cells are just non-differentiated cells. They can become literally anything. So once the notochord has developed into that right and left, there's a bunch of other proteins and genes that get activated that tell certain cells to do certain things, but that's deep into molecular biology and we're not going to get into it, okay? Just know that cells nearby other cells can influence what they become, okay? That's why your heart cells are all heart cells, and your lungs, while nearby, are not heart cells, okay? Somites. So this gets into the mesoderm. Before we were talking ectoderm, now we're talking mesoderm. So the mesoderm starts as kind of this paraaxial mesoderm, okay? So remember that I drew that notochord, and that notochord goes all the way up and all the way down, right? Our somites, the paraaxial somites, they are just little guys on the side. They help to divide your body into blocks, into kind of like, if you've ever seen an image of like the lines that people put on bodies, like There's lines that go for surgery, like surgical lines that go with the grain and against the grain. You kind of know what I'm talking about there. This is the formation of those with the grain and against the grain. Does that make sense? Kinda. It'll make more sense later. Don't worry. Just know that you have those paraxial mesoderm, and they divide into like 40 blocks called somites, and then you'll have these all the way up and all the way down. Next to those somites are the intermediate mesoderm. They're a little bigger, but they're continuous to that paraxial mesoderm. Okay? And then eventually you will get to the lateral plate mesoderm. And we'll talk about that. Okay? Bless you. So this lateral plate, the lateral plate is the most off to the side part of the mesoderm. It stays unsegmented, but it does split into two layers, okay? The coleum is what we call the space in between the two layers. Don't worry about that too much. The somatic mesoderm, so the body mesoderm, is next to the ectoderm. Then the splanchnic, I'm putting it down here because it doesn't like that spot apparently. Splanchnic mesoderm is next to the endoderm. So more on the internal organ side. Okay? Just didn't want to be there. But that's what that is supposed to be. Cool? Cool. The verbal, or the visual of all of the things that I've just said. Okay? I find that this can still be a little bit complicated to look at, but this is that neural plate that's beginning to form. Okay? So we go plate, groove, and if I could flip my hands inside out and backwards, I would, to go tube. So plate, groove, tube. So now we're in the groove part. That neural groove has formed. These neural crest cells on the side will eventually become those sensory nerves, all that stuff, until you form that neural tube. And then that fully gets locked out, done. We'll grow upwards towards the head and downwards towards the tail. It folds. It folds. You are a tube within a tube. This is the outer tube. This is the inner tube. And there's tubes everywhere. It's tubular time. Totally tubular, dude. Cool? Cool. Okay, so I think that pops off. Nope, it doesn't. Just kidding. I did bum rush these into one PowerPoint, but it was actually 3. Okay, so that's week 3. A lot of stuff happens in week 3, right? It's a pretty busy time. All that neurulation that you've got going on, all the differentiation that you're starting to do, you're starting to form into your tadpole era, right? You still have a little bit of a tail. You still kind of look like a fish. Tadpole era, okay? Right, embryonic disc bulges, week 4. yolk sac starts to grow relatively quickly. The primitive gut starts to form. Good. It doesn't really start doing anything just yet, but it does start to kind of close off the tubular part of the yolk sac. It will eventually be where your digestive and respiratory structures show up. But it's going to take some time. Okay. So, derivatives of the germ layers. We talked about these just a little bit. The ectoderm is what will form your brain, spinal cord, and the epidermis. There's technically 5 layers of your epidermis, but we'll get into that when we get into the integumentary system. Okay. Your neural crest cells, we talked about those a little bit. They will form some of your sensory nerves. They will also form your melanocytes. Your melanocytes are what give your skin pigment. And some of the bones of your face. Okay. The endoderm ecto versus endo. Don't get those confused. The endoderm will form the epithelial lining to your gut, as well as the tubes for your respiratory system, your digestive organs, and your bladder, your urinary bladder. Do we remember what type of tissue are your... Okay, so ectoderm, outer body, endoderm, inner body, and the third layer, the mesoderm, is quite complex. It is different from all the other layers. I mean, we just talked about all the stuff that gets formed by the ectoderm, all the stuff that gets formed by the endoderm. There's more than just your brain and spine, skin, and respiratory organs inside your body. Your mesoderm is everything else. Okay? So everything else in the middle is your mesoderm. The notochord would... No, it's not. Yeah, it is. The notochord is a segment of the endoderm. Kind of, not really. I move stuff around. Yeah. The notochord is kind of a part of the mesoderm, but it's also kind of part of the ectoderm. Don't think about it too hard, okay? Just know that it exists. And that will eventually give rise to the nucleus pulposa and the intervertebral discs. The nucleus pulposa is the kind of spongy bit that allows for your spine, or your spinal column, the bones itself, to have a little bit of flexibility and give to them, so that when you jump on a trampoline, you don't crush your spine just from like the weight of gravity. Okay, so that nucleus pulposa and the intervertebral discs help to kind of keep your spine in check. And we'll get into that when we talk about bones. Okay, cool. The mesoderm, that mesoderm, the segmented part of it, turns into the sclerotome, the dermatome, bless you, and the myotome. Okay. The sclerotome will form your vertebrae and ribs. The dermatome will form the dorsal dermis. So the back layer, how do I say that? The layer on the back side of your body underneath your epidermis. So there's your epidermis and then your dermis. So it forms the dermis in your back. Okay, and then the myotome will form your limb buds, which will eventually become your arms and legs, and the segmented trunk muscles. So like your abs, your pectorals, your what are these guys? I know what they are. You don't know what they are yet. Your serratus anteriors, all of that kind of stuff. Okay. Your intermediate mesoderm will form your kidneys and your gonads. Okay. And then your unsegmented mesoderm, remember we have the segmented part and the unsegmented part, but the unsegmented part does split, right? Remember that? The splanchnic mesoderm will form most of your musculature, some of your connective tissues, the serosa, remember those two layers, the parietal and the visceral and the parietal layers of your respiratory and digestive structures, your heart, most of your blood vessels, all that stuff. Okay? And then the somatic mesoderm will form the dermis, so the underlying layer of your skin, your bones, and your ligaments. So the mesoderm is doing a lot. It's pulling a lot of weight. Okay? Mesoderm does quite a bit of work. Okay? There it is, the germ layers. They exist. They're cool. This is a good and useful chart to tell you what is what and what makes what. All three germ layers, ectoderm, mesoderm, and endoderm, all made from those epiblast cells, right? At some point, those epiblasts will become epi and hypo. The hypo will form the endoderm, part of the mesoderm, but remember that those hypoblast cells were once epiblast cells. They were all epiblast cells, okay? So I would take note of this. Will really help you determine what makes what. Okay? Okay. And then the last 5 to 8 weeks of the embryonic period, you get your limb buds, you start to form limbs, so that's cool. You start to look a little bit more human. I disagree, but I don't get a say in that. Where are you? Okay. Your head is disproportionately large. Like, your head is probably like, bro, your head at this point is probably like this big, and your body is like this big. Okay. But all of your major organs are in place. They just need time to grow and become things. Okay. So this is right around the end of month 2, right around the 8-week mark. This is usually around when people find out that they're pregnant. And this is usually around when you can go get an ultrasound. You can hear heartbeats and things like that at and around 8-10 weeks or so. But every fetus is a little different. Depends on how they're positioned, all that stuff. Babies are annoying. They don't always do what you want them to do. So this is a 29-day-old embryo. We have just barely started to move out of the embryonic phase into the fetal phase. This is what I call alien baby phase 1. Alien phase 1. Because I don't know about y'all, but to me that does not look like a human being. That looks like a baby alien that's waiting to be hatched. Okay? But now we're in the fetal period. Yay! In the fetal period, basically all that's happening is a lot of maturing and a lot of growth, mostly just growth. Okay? The cells are differentiating like crazy. You are getting placed into wherever they need to be. All of the heart cells that decided they wanted to be heart cells, they said, hey, come over here. I need more of you to become heart cells with me as we're developing more and more and more and more and more of these cells. Okay? And same for all of the other organs. A normal birth is anything after 38 weeks. Anything before that is considered premature. I believe the 26th week, I want to say, you can survive outside of the maternal parent's body, but like barely. At 26 weeks, the fetus it's so small. It's less than a pound. It's like, I'm not even joking. It's smaller than this can. They are so tiny, and usually at around 26 weeks, the survival rate is not super high. So you want to wait as long as you possibly can to let that alien baby cook. Okay? I don't know if it's gonna let me play this. Sometimes it does, sometimes it doesn't. When you guys have the PowerPoints on your own. Okay? All right. So, end of week 8, alien baby 1. End of the 3rd month, I call this alien baby 2. This is still kind of first trimester area, okay? Moving into month 4, about week 13, week 16, you start to look a little bit more human-like, but just a little bit. You still definitely look like a raw chicken. So this is raw chicken phase 1. Month 5, raw chicken phase 2. You look pretty much the same, but you're just gaining a little bit more fat. Some hair follicles are growing. Baby can start the mother can start feeling the baby in uterus, probably kicking, fluttering, but probably not strong enough for anyone else to feel. Okay? This is raw chicken 2. Right around month 6 and 7, that's when you start gaining a lot of weight, when you start adding all that baby fat, when you start looking a little bit more human baby-like. Still a little raw chickeny for my tastes, but are available, right? They have periods of being awake, periods of being asleep, and you, as the person carrying that fetus, you know when they're awake and you know when they're asleep. They're usually awake when you want to be sleeping, and they are asleep when you're awake and like wanting them to interact with the world in utero, but they have no business doing so. Anyways, whatever. So month 6 to 7, I like to call that the skinty baby phase. Okay. And then in month 8 and right around up until birth in month 9, you're just pretty much gaining weight, learning how to use your lungs. When you're in utero, your lungs do work, but you're not breathing air. You're just breathing in all of your amniotic fluid over and over again. You're just breathing in all of your poop and pee, because what else do you know? We'll talk about a baby heart at some point when we get to heart stuff and cardiovascular. There's actually holes in their heart that close up as soon as you are born. It's pretty cool. But for some people, those holes don't close, and that's a heart murmur. Okay? And then the last month and a half or so, I call this the getting cute era. Because you're just gaining weight and becoming a cute little baby that has squishy little baby cheeks. Okay? They're born and they still look like raw chickens, but that's okay. They're cute. All of your newborns are so cute, and they don't look like aliens or chickens. Okay? Cool.